This video is going to be on the topic of cell communication. For multicellular organisms like you and me and all animals and all plants, it's very important that all of the cells that make up the organism are able to work together because they have to function as one organism. So these cells need to be able to, quote unquote, talk to each other. They need to be able to communicate, send signals out to other cells, alter what they're doing so that the whole organism can function as one organism. So this is going to be about how cells do that. First, let's look at this idea of feedback, because feedback is really important for the process of cell communication. Cells maintain homeostasis through this process of feedback. So let's review, / maybe you're not sure, what is homeostasis? Think about that. So homeostasis is essentially like having the right balance of everything within the organism. And this is things like how hydrated, how much water is in there, the right balance of different ions, the right balance of, energy availability, having the right temperature, maintaining the right pH. So it's basically keeping all of the systems, the whole body, at the conditions it's supposed to be at. So maintaining that correct level of everything. And it's very important for the body to be able to maintain the right level of all the things in order to be able to continue to function properly. So in order to do that, it uses a process called feedback. And particularly, we're talking about negative feedback. So what occurs with negative feedback is there is a lack of homeostasis. Something has gone out of whack. It's too hot, there's too much sugar in the blood, whatever it is, something is wrong, not in homeostasis. Then there is a response by the cells, and the purpose of that response is to return the body to homeostasis. And then once homeostasis has been reached, there's sort of an automatic shutdown of that response. In contrast to negative feedback, there's a process called positive feedback. Now, positive feedback is much less common than negative feedback. Negative feedback is happening just all the time, but there are certain situations where we might want positive feedback. So positive feedback, imagine like the snowball rolling down the hill. And as it rolls down the hill, it gets bigger and bigger and bigger. So while negative feedback, we get a response that gets shut down once homeostasis has been reached. Positive feedback pushes the body further and further and further out of homeostasis, creating a larger and larger and larger response. And so on the next slides, we'll talk about examples of why you might use those. A real-life example of positive feedback outside of biology is what happens when you get a microphone too close to a speaker. Because what's going to happen is the speaker will pick up sound from the microphone and broadcast that, which will then be picked up by the microphone, which will then go into the speaker again. And so you get that horrible noise you get when a microphone gets too close to a speaker. It's sort of a bigger and bigger and bigger response. It kind of goes in a circle. So an example of positive feedback in mammals is when a woman or female goes into labor. This might be a little graphic, but what happens when a woman starts to go into labor is that a hormone called oxytocin is released. This causes the uterus to contract, which pushes the baby down. So the baby's head gets pushed down against the cervix, which stretches it. The stretching of the cervix causes the release of more oxytocin, which causes more contractions, which pushes the baby down further, which causes more oxytocin, and so on and so forth. So you get this bigger and bigger and bigger response, stronger and stronger contractions, until finally the baby is born. Another example of this might be an inflammation response. When you get, say, a cut or something and some bacteria gets in, your body will start to produce an inflammation response. And that response gets bigger and bigger and bigger until the wound or the bacteria is taken care of. Sometimes this kind of response can get out of whack, like, for example, allergies, when the body responds inappropriately to something that isn't actually a threat but causes more and more and more inflammation, sometimes to the point where the person maybe can't breathe because their throat is swelled up so much because it's so inflamed. So those are a couple examples of positive feedback. Again, negative feedback is much more common, and negative feedback is how we maintain homeostasis. So a couple examples of that, say the body temperature is too high. It's hot out, you've been exercising, whatever. This will cause the response of sweating, which should cause, through evaporative cooling, the body temperature to drop. And once the body temperature drops, the sweating stops. So it's kind of a self-shutting down process. Maybe blood sugar is too high. You just ate a bunch of sugar. This will trigger the pancreas to release insulin, which causes the blood sugar to go down. And once the blood sugar goes down, the pancreas stops releasing insulin. So these are just a couple negative feedback examples. You can take a minute for yourself, jot some down. Can you think of any other positive or negative feedback examples in humans? So now we're going to go to the actual sort of mechanics of cellular communication. There are three basic steps, three sort of categories of steps in the cellular communication process. The first step is reception. The signal gets received by the cell, so some outside signal triggers the start of this process in the cell. The next step is transduction, and the transduction process is sort of this chain reaction idea. So it's kind of like those Rube Goldberg machines, like maybe you made freshman year in physics, where this thing triggers the next thing, triggers the next thing, triggers the next thing. It's that whole chain reaction. And sometimes this chain reaction may amplify the message. For example, if you had, in the Rube Goldberg machine, you had a line of dominoes that are falling down, and then maybe it's going to spread to two lines of dominoes, so this one is going to knock down this one and that one, and now you have your signal amplified because you have sort of two lines of the signal going out. So that kind of thing can happen in the transduction process as well. And then finally we get the response, which is the final result of the cell communication, whatever the cell ends up doing in response to this communication. So you can kind of think about it as like reception's the first step, response is the last step, transduction is everything that happens in the middle. And then this is just a diagram kind of demonstrating those three steps in action. And we're going to look at those three steps in more detail now. So let's start with types of reception. Cells embedded in their cell membrane have receptor molecules, like so. And the receptor molecule will come in contact with signal molecules that will bind, perfectly fit, just like the enzymes. It's a perfect fit inside of the receptor molecule. There's different ways this can happen. Sometimes it can be through direct contact with another cell. So like what's showing in this example, this cell might have signal molecules embedded outside of it, and then this cell has the receptor, and when those two cells come together, that initiates a cell communication. This can happen, for example, with your white blood cells and your other immune cells recognizing your cells as your own. The white blood cells will actually come up and sort of grope and grab at the cells, and when they feel those signal molecules on the outside of your cell with their receptors, they'll say, okay, this cell's cool, I don't need to kill it. This can also happen when cells that are growing sort of bump up against each other, and that can signal them, okay, it's time to stop growing and dividing, we've run out of room. A lot of times, cell communication occurs not from direct cell-to-cell contact, but rather from cells releasing signal molecules through exocytosis that then travel to other cells. And this can happen with a cell just sending signal molecules out that travel to the other cells in its immediate vicinity, or it can happen with cells sending signal molecules out into the bloodstream, which travel around the body to reach their final cell. We call these long distance traveling signal molecules hormones, and the shorter distance are called local mediators. So generally, the process of reception looks like this. We have a receptor that is embedded in the cell membrane. Here's the cell membrane. And then we have our signal molecule. Now, another name for the signal molecules is ligand. A ligand is just a smaller molecule that binds to a larger molecule. So you might see either of these terms used. Generally, in most cases, the ligand doesn't actually go inside the cell. It binds to this receptor that's sticking out of the cell membrane, which causes something to happen with this part that's inside the cell membrane, usually a change of shape, and that's what initiates the signal. It doesn't actually go in, it just binds to the receptor on the outside. So since the receptors are embedded in the membrane, we call this part that's outside the membrane where the ligand actually binds the extracellular domain. So extra is like outside. And we call this part that is sticking inside of the cell the intracellular domain. So what usually happens is the ligand binds to the extracellular domain, and this causes some kind of change to the intracellular domain. So we're talking like some kind of shape change, but it could trigger something else. And this is what begins the transduction process. So this is like the first domino tipping over in the Rube Goldberg machine. So here's a couple specific examples of receptor types. One receptor type is called an ion channel receptor. So these are those passive transport facilitated diffusion proteins, ion channels. What happens is the messenger, the ligand, whatever, comes and binds in the ion channel, and that causes the ion channel to open up and start allowing ions to flow through it. So normally the channel is closed, but when the ligand comes and binds to it, it causes it to open up. Another type that's a little more complicated, and honestly, these cell communication things can be pretty complicated. There can be a lot of steps and things happening, but another type is called a G protein receptor. And so here's the G protein receptor. The G protein receptor has inside the cell this protein called a G protein kind of attached to it. So the signal molecule comes, the ligand, and binds to the G protein receptor. This triggers the G protein to be phosphorylated by a molecule called GTP, which is really similar to ATP. So that sort of provides energy to it, and it causes it to move over to an enzyme, a different protein, bond with it, and then that enzyme is going to initiate the transduction process, so a new chain response. After it's done this, the GTP comes off, the G protein goes back to its receptor, and it can be triggered again if a new signal molecule comes in. So it kind of goes back and forth. This is sort of like an on switch or an off switch. If the signal molecule is here, it can go and activate its enzyme. If it's not, then it doesn't. Here are some review questions for you to consider for yourself. I know this is kind of a longer one, so let's pause and review. So that's what reception looks like. Let's move on to some of the things that can happen during transduction. And again, transduction can be really complicated. There can be a lot of different things happening, molecules that have names that you're not familiar with, and this thing becomes this thing, activates this thing, activates that thing. It's just this long sort of chain reaction again. The important thing is to just kind of try to follow through what's happening. Okay, this activates that, that activates that, that activates that, and not get too caught up in the names of all of the things. One thing that can happen during transduction is phosphorylation of proteins. So remember, we've got like ATP, adenine, and 3 phosphates, and this is the energy molecule for the cell. And the way this energy molecule works is the energy is stored right here, and when it's used, one of those phosphates, the last one, pops off, and it sort of gets attached to whatever the thing is that it's energizing or activating. So a protein kinase is an enzyme that phosphorylates another protein. So a protein kinase is the enzyme that sort of does this process of activating, energizing, or phosphorylating another protein. So usually what happens is when the phosphate gets attached to its new protein and provides it energy, this causes a change of shape, which then allows that protein to go and trigger something else, do something else. And what can happen with this phosphorylation process is we might have one protein that phosphorylates the next protein, which phosphorylates the next protein, which then phosphorylates the next protein. We call this a phosphorylation cascade. So it's literally kind of like, again, the dominoes being knocked over one after another. And so here is a picture of that process. So signal molecule, ligand comes in, binds to the receptor. The receptor phosphorylates sort of this first protein kinase, which then phosphorylates the next one, and then the next one, and then the next one, sort of all the way down the line until we finally get to whatever end cellular response we have. Another common thing to see in the transduction process is something called second messengers. Remember when I talked about how one domino maybe knocks down 2 different dominoes, and then our signal is amplified. So second messengers, the name, they are not necessarily like the second step in the transduction process. That's not what that means. We just call them second messengers. And what happens with second messengers is basically some part of this transduction process triggers a release of a bunch of small molecules out into the cytoplasm of the cell. And then these small molecules can each go and kind of trigger another molecule and activate the next thing. So the signal gets amplified from one ligand coming in to this whole flood of little molecules going into the cell, and each one of them triggering a response. So it's a signal amplification process. Second messengers are small molecules, sometimes just ions, for example, calcium ions or potassium ions. There's also another one you might see called CAMP, which is called cyclic AMP. The point is they're small things. So if we look at, for example, this whole signal transduction process, we have again our ligand, our signal molecule. It binds to a G protein receptor. The G protein becomes phosphorylated with GTP, which activates this enzyme, phospholipase C. The phospholipase C breaks up this molecule into this DAG thing and this IP3. The IP3 ends up binding to an endoplasmic reticulum, which is full of calcium ions, causes the massive release of calcium ions into the cytoplasm. So the calcium ions in this example were a second messenger, and this can cause various proteins to be activated. So they might activate a bunch of the same kind of protein, or they might activate a whole bunch of different proteins, and now we can have like 3 different responses going. And then finally we get our cellular response. So this is a picture of kind of what a whole transduction pathway might look like. Again, complicated, a lot of steps, but if you kind of just break down, okay, this does this, this does this, this does this, it's not, too bad. Okay, here are some review questions again. These 1 and 2, so you can pause and take a look at those, but I do want to talk about 3 as well. This is something that we haven't addressed yet, but that final phase of the cell communication response. So what are some things that a cell might do as a response? Right, so some examples of things a cell might do as a response, like literally anything a cell could do. Maybe the cell divides as a response, makes more cells. Maybe the cell dies as a response. Maybe that was a signal telling that cell that it was no longer needed. Maybe the cell turns on a certain gene, or maybe it turns off a certain gene. Maybe it causes it to produce or stop producing a certain protein. Maybe it causes that cell to, move somewhere else. Maybe it causes it to do endocytosis and take in something from the outside. Maybe it causes it to release something. So there are just a ton of different things that could be the result of cell communication. Again, basically anything a cell could do, it might be signaled to do through this process of cell communication.